Can you isolate all the bacteriophages in the world? No. Even if you do isolate, the bacteria is evolving and the bacteriophages are also evolving. Then how do you catch up to those, let's say, evolutions altogether? It's insane, right? We start with the matching and maybe putting the bacteriophages in practice, etc. But in cases where you do not find any bacteriophage or there's no antibiotic that can be used, then what if we could engineer new bacteriophages in silico that can be then taken into production?
Welcome back to the Smart Biotech Scientist. I'm David Brühlmann, your host, and this is part 2 with José Bila, who is the CEO of Precise Health, where we're exploring the business and regulatory sides of AI-powered phage therapy. In part 1, we've heard his compelling personal story and learned how artificial intelligence can revolutionize treatment selection against bacterial infections. Now, we're diving into the nitty-gritty, the production scalability, the regulatory hurdles, and the economic realities of bringing personalized phage treatments to market. Discover how AI is making life-saving therapies both accessible and affordable for patients worldwide. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brühlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. How do you produce these bacteriophages, and also how long would it take? For instance, in this case, that's the unfortunate event. We have 80 or 90%, but unfortunately a patient comes with that 10% we have not covered. How fast could you start bioreactor, and then how long will it take? And can you tell us a bit what kind of bioreactors we're using to produce bacteriophages?
The production is actually, it's quite complex by itself, but to be honest, it's not the most complex thing, and it's been done like for ages. I'm not bioprocessing person, but I would reckon that it's a little bit less complex than a chemical molecule that you have to do synthesis and then everything else. So it's quite less complex, I'll say. And the type of bioreactors are standard bioreactors. So the same you use for, let's say, bacterial growth and so on is the same ones that you use for bacteriophage growth and production. Now, in terms of timing, actually, depending on who is doing the production. So some hospitals already have their own capabilities of producing them in-house, which is a major gain. Most hospitals, they don't have that. So you need to go to a commercial CDMO. And when you talk to commercial CDMOs, the least can actually do that is somewhere between 2 to 4 weeks of time. And that's assuming that when you give them the mandate, they will have an empty bioreactor and the capabilities of personnel to actually start doing the production for you. And then there is another issue, which is, say, stability, quality control, because you need to go over all of these steps in time. Everything needs to be considered, essentially. It's the classic, say, GMP process that needs to be taken into consideration. And all the quality management system and so on need to be in place in order to be able to do that more efficiently and correctly, so to speak.
I guess a dual system could work very well. You could use a CDMO to produce your 80-90% bacteriophages. You need over and over again. And then you could have in a few hospitals some smaller bioreactors to quickly produce whatever is lacking. That could be a solution.
And this is correct, because the reality today is that only one hospital in Switzerland that we know of, or I know of, actually has the capability to produce in GMP. The rest of the hospitals in the world, they don't have GMP capabilities. However, when they need to use phages, or they see the need to use phages in their patients, they have the processes in place which are not GMP but could be GMP-like, or the right, let's say, sterilization methods, etc., that could bring the bacteriophage to the patient. So some hospitals have already invested in that kind of capability. But again, that's a big assumption that most hospitals in the world will be able to do that. I don't think it's correct. In Europe, yes, many big hospitals, most likely university hospitals, may have that capability. But when you go to regional hospitals, this is, I think, quite difficult. US, maybe yes, in the big hospitals, again, bigger cities, bigger, well-funded hospitals. But in rural areas, this is not going to happen as swiftly as we want. And we also have to account that most antimicrobial resistance cases are coming from LMICs. So this is low, middle income countries. We have whatever strategy we develop today, we need to take that into account, because this is where the most impact will be made altogether.
Could we envision, let's say, some mobile facilities somewhere in a remote area of Africa or Southeast Asia? Because, yeah, as you mentioned, that's where the most cases will be. I think in the Western world, we could cover it with a few centers and then quickly ship it if needed. But where it will take time is especially in those remote areas. And probably that's also where the expertise is still lacking today. So how could we go about producing that in a very economic way, in a cheap way, maybe with single-use technology or some mobile reactors? Or, I mean, we probably need some creativity there.
Yeah, exactly. I think if we manage to build a single-use bioreactor for each patient that is cost-effective some way, that will be $1 billion idea for sure, because this is something that I think all personalized approaches would need somehow. And the issue, I feel, is not really on the bioreactor itself. It's really on the downstream processing of things, because you want to be able to build a bioreactor, but you need to combine it with they say downstream processing in a way that you get something that is pure. But how do you miniaturize it in a way that is going to be cost effective? I mean, if anyone has some ideas, we are happy to collaborate on those things, because yeah, this is $1 billion idea for sure. But for the LMICs, the way I see it is that, I mean, of course, we are in Switzerland. We are fortunate enough to have these big hospitals that make us think outside the box initially, without the thinking of the costs or something. We can already kind of start implementing, say, thinking of ideas of how to make it work. And then in a second phase, we will have to be able to decrease the cost so that we can feed everybody else who does not have the same economic power. Now, for LMICs, I think we will have to implement it here first, make sure that it works. And then how do we go down the line? That's going to take a bit of time. But I am from LMIC myself. And like I said, my parents died from the antimicrobial resistance. And in that case, even if we had bacteriophages today that could have saved them, I wouldn't have benefited them by just focusing on financially powerful markets. So I need also to think of that. And this is something that attracts me to my team every day. Whatever you do, think long term, 3 to 5 years from now. If you were in Uganda, for example, how would this be used and how would it be implemented in a hospital where it does not have the same capability as one big hospital in Switzerland?
Yes, that's a very good point, Jose. Start with the end in mind, because finally it's about the end user and how are we going to bring it to them. Even if we have the best technology, but we cannot deliver it or it's too expensive, it will be of no use. Now, I want to double down a bit on the process again. So since you mentioned the downstream, can you tell us how different the downstream is from a standard, let's say, MAP process or like a viral vector process? Is that very similar or is it very different?
From what I understand, it's quite similar. I'm not the one doing this on a day-to-day, and I'm not very familiar with the bioprocessing of viral vectors, but the phages are viruses themselves, so I wouldn't expect something that is abruptly different, I'll say so, in that case. But I think maybe one thing that we can think of, which I mentioned earlier, is the question of stability. This is something that is very, very important. Let me take it back a bit and say some doctors, of course, they're talking about logistics. Okay, how do I get phages, the right time, the right one, etc., etc. This is one issue. And then the second issue is the lack of clinical data and so on and so on. We have enough cases that have been treated, but it hasn't followed the systematic, let's say, classic clinical trial approach so that you can use the same data points to implement into that. So this is one thing. And then on top of that, there's been some clinical trials done back, I think it was 2017, if I'm not wrong, on phage therapy. And there, one of the things that came up was that they had this cocktail of phages, and it was a 10 to 6 PFUs, and that's how they wanted to use that on wound infections. And with time, what happened is that they saw that the stability was not great. Instead of administering 10 to 6 PFUs on one patient, they ended up administering 10 to 2 PFUs per milliliter. So this is not a lot. This is not great. One issue again there is the stability. And then the second thing, maybe was a problem in this specific clinical trial, was the fact that you need to test each patient bacteria isolate against the cocktail. And before you actually administer the cocktail, because you are just assuming that the cocktail is the magic potion that is going to treat everything just the same way you're doing with antibiotic. So this is one issue. But again, coming back to the issue with the stability, if we want to implement our strategy, which I discussed to you, the case of taking, let's say, bacterial fingerprint of a specific region and so on. If you want to stock the phages, you need to be able to keep them for long. That's how the economics are going to go. Because if I'm having a batch that is going to stay only for 3 months, then how likely within 3 months am I going to find patients that are going to utilize that specific batch, right? That's number one. If we are able to increase the time, I don't know, stability, shelf life to 2 years, 3 years, whatever, if you can, then that's where the economics are going to make a lot more sense, because I can produce whatever I want and I just keep it there. Eventually, someone is going to use it at some point, right? So that's how we think. And this is where miniaturizing, like coming again to your idea of miniaturizing the bioreactor that can do one batch approach and giving it to the patient. Until we get to that, we need to be able to get proper stability of phages. And one way to do that, of course, is through these lyophilization approaches that different people have been working on and have been looking into to increase stability.
That's a very important point. Before we continue, biotech founders, I want to talk directly to you. Do you know that feeling when an investor asks about your manufacturing strategy and you freeze? One wrong CMC decision can cost you $2 million and 18 plus months of delays. Yet many CEOs are flying blind on these critical decisions. That's why I created the 1-week CMC strategy assessment. In 1 week, you get a personalized assessment of your molecule and development stage. We identify critical gaps before they become million-dollar mistakes, plus a strategic roadmap for high-stakes investor meetings. One CEO told me, David took me from feeling powerless about our CMC roadmap to confidently pitching investors. An absolute game changer for time-pressed CEOs. Whether you are preparing for Series A, approaching IND filing, or facing scale-up challenges, this gives you confidence to ace investor and board conversations instead of surviving them. Ready to stop second-guessing your CMC strategy? Check the link in the show notes and secure your spot. Because your breakthrough therapy deserves a solid CMC strategy. Now, let's get back to our episode. Right, Jose. I just want to also briefly touch upon the regulatory aspect, because finally it's a drug you're going to be used to treat patients. How do you navigate this with the health authorities, especially as you're using machine learning and AI approaches to identify the phage? Is that relevant or not? Or how do you go about that?
There are two fronts of regulatory, and this is always, regardless of which investor we talk to, it's always the big problem. So what is the regulation around this? So there are two fronts. One is the phage therapy by itself. So if I'm a doctor, I want to administer phage therapy, then which set of regulations should I go by? And then the other one is the process to identify the right phages and then get them produced before they can be used in the first, say, front of regulatory aspects. So the first one, these many countries, and I think if you're very deep into the phage world, you know that a lot of different stakeholders are talking about finding the easiest way to implement this into the hospitals. So Belgium, for example, they use these magistral preparations for each patient case. So they don't go through GMP, for example. So they waive the need for GMP. They prepare them in the hospital and then they administer them to the patient as they go. So they have a good library of bacteriophages, and whenever a doctor needs them, they still go through this manual process that I just described earlier of identifying them, and then they produce them and administer. Last year, I think around November 2024, Portugal said, okay, we are following suit. So we can also follow the same model as Belgium, as long as there is a clear need for the patient and there's no other alternative. Let's use bacteriophages. So doctors can use bacteriophages as long as there's ethical approval within the hospital committee, etc., etc. So that's one way. There is some relaxation, and we know that some other countries in Europe, more specifically, are following the same model. We have some stakeholders outside Europe as well that are following the same model. So there's some kind of relaxation around there. But of course, the European Union has, in the wide, they still need more clinical trials in order to be more certain about this altogether. Now, on the second front, which is the use of machine learning to do the identification, this is also still not mature enough to get accepted directly, but we have some precedents. The same way you have antibiograms in hospitals, the processes of antibiogram in the hospital is the same thing. Just think that we are not testing for antibiotics, we are testing for phages. If you were to have a digital antibiogram, why don't you have a digital phagogram in this case? So there is precedent into that we can use, and we are not trying to build a platform that we are just going to go wild and just use it however we want and tell each doctor just use it. We are actually also aiming to follow suit with CE marking and all the regulatory approvals necessary for that. And we are putting everything in place to actually get there. So this is also building confidence for the medical doctors to use it, but it's also building confidence in us because we are following all the regulatory pathways in order to get this used in clinical applications.
What is your vision for the future of this machine learning powered phage therapy?
It's a deep question.
Yes.
So I think there are many fronts to that, but we believe that today most natural entryway to the clinics in Europe or anywhere in the world that they are not using phages today is to use natural phages, for example. So you extract them, isolate them from multiple sources and so on. And if you're isolating so many, then identifying the right ones is going to be the issue. Reducing the logistics, etc., is going to be the issue. So that's where we think machine learning, it can be used first. Most importantly, if I think 3 to 5, maybe even to 10 years from now, that I think is where we will have the most impact. However, we have to think further. What about the cases? I think the estimations was that on Earth, there are 10 to 30, say, bacteriophages. Somewhere around there, the number, I remember it was 30, so forgive me if I'm not saying the correct number, but it's a huge number, right? So if you think in that way, can you isolate all the bacteriophages in the world? No. Even if you do isolate, the bacteria is evolving and the bacteriophage is also evolving. Then how do you catch up to those, let's say, evolutions altogether? It's insane, right? We start with the matching and maybe putting the bacteriophages in practice, etc. But in cases where you do not find any bacteriophage or there's no antibiotic that can be used, then what if we could engineer new bacteriophages in silico that can be then taken into production? And this is also the vision of Precise Health, because this is what we want to implement, a system where you can match first from the existing libraries. And if you don't find anything, then can you, in one click, just engineer new phages? And with those phages, can you then do the production and potentially use in a patient and so on? Of course, it's so much complex than what I'm saying, and it's going to take time. This is why implementing the engineered ones is a little bit harder and it's going to take a bit more time. And that's why we're starting with the matching.
Wow, the future definitely looks bright or interesting at the least.
Yeah, true.
This has been great, José. Before we wrap up, what burning question haven't I asked that you are eager to share with our biotech community?
Burning question. So it's not really a burning question, but it's more of a vision for me in the sense that bacteriophages have been there for 100 years. I've said it multiple times. It's not a question of whether the bacteriophages work or not, because we know they do. They've been used multiple times. It's a question of building an infrastructure and a foundation that makes sure that bacteriophages can be implemented in the cost-effective way, in an accessible way altogether, so that we have something that is more global and thinking of the whole, not just the Europe, but across the globe altogether. And we think machine learning approaches are the solution for this. And this is what we are trying to show to the world and everybody else. So maybe not a burning question, but burning comment, I'll say so.
Fantastic, José. With all that what we've covered today, what is the number one takeaway you want our listeners to walk away with?
We don't need to wait too long to find solutions for people who are dying, because this can affect anybody. Can be us, like I said, could be your loved ones and so on. And antimicrobial resistance is one of those things currently. And whatever systems we have today don't work. So if you have an idea how to solve that, or even if it's not in the MR space, anything else, just go out and try to do it. And eventually someone will find the right solutions. I think that is a key takeaway, I guess, all the listeners who are out there. Thank you so much for listening to our troubles, I'll say.
Yeah, thank you so much for moving the needle, José, and thank you so much for investing your time and passion to solve a real problem of humanity. Yeah, also thank you very much for coming on the show. It has been a huge pleasure to talk with you. Before we go, where can people connect with you?
We can be found at precisehealth.io. That's our website. But for those who want to reach me directly, it's jose.bila, B-I-L-A, at precisehealth.io. So I'm happy to connect with anyone who might have questions, or I can help with anything.
Fantastic. I will leave the links in the show notes. So reach out to José. And José, once again, thank you very much for coming on the show. It has been a huge pleasure.
Thank you so much. It was a pleasure to me. My first podcast. I hope it's impactful to anyone who is listening. Thank you so much.
José's vision for AI-powered phage therapy offers real hope in our fight against antibiotic resistance. His journey from personal loss to breakthrough innovation proved that biotech can truly save lives. Do you have questions about process development or manufacturing challenges? Well, book a free consultation with me at Brühlmann-Consulting/call, and I'm happy to help you get started. And please rate us on Apple Podcasts or your favorite platform, because every review helps us reach more biotech scientists ready to make their mark. Thank you so much for tuning in today, and I'll see you next time. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.